Hello, dear students. We are here again with microbial anatomy and physiology. And here we have to discuss a DNA replication. And before this, if you remember, we have talked about the synthesis of components in cell wall for its replication. How cell wall elongates, how it divides into two, and other components are how going to be divided into two from one bacterial cell for replication. And here we have to talk about DNA replication. We know very well DNA is the major structure or organelle in bacterial cells, which is present in cytoplasmic region of bacterial cells. And DNA is responsible to control all characteristics of microorganism, all functions of microorganisms through gene. So it is very necessary and important. It must have been divided into two in form of copy to transfer one copy into daughter cell. That same characteristics will be transferred from one bacterial cell to daughter bacterial cell. So DNA replication, we have discussed model of DNA replication and Melson's still experiment we will discuss in it. DNA synthesis and elongation, we will talk about this. And enzymes which are responsible for DNA replication, that is, those are DNA polymerases. We will discuss which type of enzymes are responsible in DNA replication. Origin and initiation of DNA replication, we will discuss about this. Prokaryote and eukaryote models, circular oblique linear chromosomes, telomerase replication, histone and chromatin assembly. We will discuss all these things in this chapter. So first one, we have to discuss the alternative model of the DNA replication. There are proposed three models or three ways which has been assumed in DNA replication, that is semi-conservative model number one, and second is the conservative model and dispersive model. These three models we will talk about how replicates. Suppose in semi-conservative model, this is the parent DNA which have two strands of DNA. When it is going to be replicate, according this model, semi-conservative model, each strain will work as a parent strain and the daughter strain will synthesize on it. Suppose these are offspring, or these are two replicates. This is parent strain of DNA and this one is a daughter strain of DNA. Similarly here, this one is parent strain which has been taken from the parent material, parent cell, and worked as template, and the daughter strain has been formed on it in first generation. So the DNA one is from parent strain which worked as a template and the daughter strain formed on it. So how? This has been opened and helicals and it becomes straight. It provides, it was available in form of template for daughter strain during replication, and the daughter strain formed on parent strain. So this is called semi-conservative. Again, this one will go in replication for new generation. So this one will work as a parent strain and will provide as a template while daughter strain will form. This one also work as a parent strain as a template and a daughter strain will synthesis on it. And this one will work as parent strain and another daughter strain will work on it. Similar. So this type of conservation in which one template is one strain is from parent and daughter strain will form on parent's material or parent DNA. This is called semi-conservative model. It is most accepted and proved model which is considered the most acceptable and valuable, which is throughout world it has been accepted. In conservative model, what they are saying that parental DNA will work as a template, and here daughter DNA will form on parent DNA. So parent DNA will be separate from it, and daughter DNA will be separate from it. Again, this one will work as a parent strain and will provide a template and the daughter DNAs will form. One is parent DNA and another is daughter DNA. Similarly, over here, DNA works as a template. It provides template where daughter DNA forms and it will be separate at last in second generation. The dispersive model is explained this. Suppose this is parental DNA, and here some segment will be from parental material, and some segment will synthesize from daughter strain, some parent. As so, it is a mixed model. So this is called a mixed model, second generation. But this model could not get placed, could not. Is not so famous. So it is not too much acceptable. This was the dispersive model. Dispersive model is not works too much. So these are alternative models of DNA replication. These are three models: semi-conservative model, conservative model, and dispersive model. This dispersive model uh, is could not get so much place in science, but semi-conservative model got the most uh, appreciable place in science. So in 1958, Matthew Malson and Frank Stell's experiment. This experiment was on equilibrium density gradient centrifugation. They have taken DNA in 6 molar cesium chloride, and they have labeled both strains with the different markers N4, N14, and N15. And this DNA was centrifugated for 50 to 60 hours at 100,000 G, results in generation of gradient of CSCl and bending of DNA. So what happened? Increasing density, suppose after centrifugation N15 was higher and it was settled down, and N14 was lighter, these two strands were separated. Here, what they did here, two strands of DNA were labeled with N14 and N15. Blue one is N14 and pink one is N15, and they separated both strands in this way. what they did from here, 1958, Matthew Nelson and Frank's experiment, semi-conservative model of DNA replication was proved by experiment. They have taken E. coli culture, that is called starter culture, start, and 15 containing medium. They grow E. coli in nitrogen 15 containing medium, and the DNA was extracted from here. DNA is cesium chloride gradient. DNA composition, DNA have heavy N15 oblique N15 DNA on both strands. So after gel, uh, because this DNA was run on gel, and after that photograph of DNA bands was taken, there was single band. And densitometric scan also showed there is one peak. Okay. Secondly, what they do? Continuous growing first generation in 14 medium. They have taken E. coli culture and they have grown in it and 14 medium replication cycle 2 from here, because these both strands were containing N15. Culture has been taken from here because this culture is uh, contains only parent material. Parent E. coli are here. So this culture was again grown in N14 medium. So what will do? If you will listen over here, the mechanism of this experiment of this stage, because the parents were using N15 and it was in DNA N15 was binded. So this culture again has been grown in another medium which contains N14. So for growth they will use N14 medium. After growth DNA was extracted from there. So what happened? Both strands of this generation have two strands, because one is daughter strand and second is one is parent strand, second is daughter strand, heavy and light hybrid, and 15 oblique and 14 DNA was present. When it was run in gel, agarose gel, it showed bands, and in densitometer metric it also showed one peak. When this was grown in replication cycle 3, it was grown again in N15, in N14. And after growth, DNA was extracted from here and centrifuged. Both N14 and N15 were separated. So what happened in this stage? Daughter strands have N14 and N15. One daughter strand have N15 and one daughter strand have and one daughter is one DNA, one generation, one bacterial cell, one DNA have N14 and N15, and another daughter strand have N14 and N14. What strands contain N14? Because this worked as a daughter strand, because this blue one was N14, so it provided a template and N14 has been formed on it, and this pink one was N15 and this N15 provided a template, and on it N14 was established because medium contained N14. And when When it was run in gel, it showed two bands. And in graph also they showed two peaks. So it indicates there is two type of strands which are this supports for the semi-conservative model that one strand contain N14 and another strand contain N50. After this, again it was grown in 14 in replication cycle 4, and the DNA was extracted and centrifuged in cesium chloride. DNA was extracted, and whence one peak was reduced, another was increased because continuously it was growing in N14, so the peak of N14 was increased, and N15 because it was parent material. Once it has been grown in N15, so its peak was reducing gradually. So this experiment proved that the replication model in bacterial DNA is semi-conservative, where parent strain is working as a template which provides a template for daughter DNA synthesis. In 1955, Arthur Kornberg, who was working with E. coli because E. coli is left microorganism, discovered the mechanism of DNA synthesis. This is the guy who was working in E. coli, and he did work in replication in synthesis of the bacterial DNA, and he found four components are required for DNA replication. Four things. We know very well that in DNA there are four bricks, four units: adenine, guanine, thymine, cytosine. Adenine, thymine, guanine, and cytosine, which are called DNTPs. These four units are working as a basic units of the DNA. Four components are required for DNA replication. One, deoxyribonucleoside 5 triphosphate, which are composed of, which have sugar molecule as a base, and phosphate molecule is present at third carbon atom. DNA template, which provides by parent material, parent DNA, and DNA polymerase, Kornberg enzymes, which are working, which are responsible to replicate the DNA into daughter strand, and magnesium, optimized DNA polymerase activity. These four units are required for the synthesis of the bacterial DNA. And this work was done in 1955. Therefore, we are saying that microbial science, science of microbiology is very new. Still, it have many things to do, to explore and to use. So Kornberg, who was working with E. coli, he discovered the mechanism of DNA synthesis, and according to him, there are four things are required. First one, basic units, basic blocks, which are DNTPs, adenine, thymine, guanine, and cytosine. DNTPs, DNA template, which will be taken, which is used from the parent, and a DNA polymerase enzymes, and magnesium for the optimization of polymerase activity. So For DNA synthesis, for DNA replication, four things are required. DNTPs, four DNTPs, adenine, guanine, thymine, and cytosine, DNA template, and a set of enzymes which are called DNA polymerases, and magnesium. When these all things will be kept in one experiment or in one solution, and the conditions will be optimized and the conditions will be adjusted, so daughter DNA will be formed from the parent DNA. So how we can do this thing? Suppose we will extract a DNA from a bacterial cell. Okay, this bacterial DNA will work as a parent DNA or template DNA. Further, we will add DNTPs, polymerases, and magnesium, and we will start the reaction. And after completion of cycles, if you will check, the DNA quantity will be double, triple, it will increase. 1959, Arthur Kornberg, Stanford University, and Estero Osho, Ochoa, these guys proved this thing, and they got this work. They dedicated this. They did this work to their name. Three main features of the DNA synthesis reaction. DNA polymerase 1 catalyzes formation of the phosphodiester bond between 3' hydroxyl of the deoxyribose on the least nucleotide and the 5' phosphate of the DNTPs. So how it works? In picture we will study it in detail, in clearly. Here just we have to understand how DNA synthesis, daughter DNA synthesis takes place. DNA polymerase 1 catalyzes formation of the phosphodiester bond between one nucleotide to another nucleotide, between 3-hydroxyl of the deoxyribose sugar on the last nucleotide and 5' phosphate of the DNTP. So DNTP, deoxyribonucleotide phosphate, it have 5' which is available for binding, and this 5' of the NTP will bind with 3-hydroxyl group of the existing or last nucleotide of the synthesizing daughter DNA. Energy for this reaction is derived from the release of two of the three phosphate of DNTPs. So this reaction is required where suppose last nucleotide which has been binded with the with the DNA parent strain. So next nucleotide molecule will come and it will bind from 3-hydroxyl side of uh, a 3-hydroxyl side of the existing nucleotide with 5' phosphate of the coming DNTP nucleotide. So for this energy is required, and this energy is obtained from the phosphate which is present over here. DNA polymerase DNA polymerase finds the correct complementary DNTP at each step in the lengthening process. When new DNA molecules are binding with growing DNA. So here this process is monitored by DNA polymerase enzymes which are making sure that correct DNTP will bind with existing nucleotide. Suppose just we are keeping one brick at bottom and the next brick we are deciding accordingly. Here also one DNTP which is binded with growing chain, growing DNA. So which the which next TNTP will come over here, it will be decided by DNA polymerase enzyme, and in this way DNTPs will come, and accordingly these will bind by 5 prime to 3 hydroxyl site of existing DNTP, and this chain will grow. The rate of synthesis is 800 DNTPs will bind with growing DNA strain in 1 second, and low error rate. It have low error rate. Direction of synthesis is 5 prime to 3 prime. 5 prime will bind with 3 hydroxyl prime, and again it will keep 3 prime open, where 5 prime will come and will bind it. DNA polymerase image created protein data bank. DNA elongation. So here we can see in detail. So this thing we will talk in next class. Uh, so here I will explain little bit to you. Suppose this is one strand of DNA. These are nucleotides: adenine, cytosine, adenine, thymine, cytosine. Okay, and we know very well this thing that adenine will bind with thymine, cytosine will bind with guanine, and adenine will bind with thymine. If parent material, this is parent DNA. Okay, this is parent DNA. This one, and new nucleotides will come over here. If there is adenine, thymine will bind. If guanine is there in parent DNA, cytosine is there in parent DNA, guanine will bind. If adenine is there in parent DNA, thymine will bind. And what will do? Second, here we will see that 3 hydroxyl group is empty. Here new nucleotide will come and will bind with it, and these will form a bond. This is bond in this is phosphodiester bond. This is phosphodiester bond in parent DNA. This is template. This is template in template phosphodiester bond, and these are nucleotide bases which are present on parent DNA. In daughter DNA, that if adenine is there, thymine will bind. Okay, thymine's 3 hydroxyl group will be empty over here. So guanine will come and guanine will bind with this through phosphodiester bond by 5 prime, and again 3 prime will be empty, and if adenine is there, thymine will come, and 5 prime site of thymine will bind with this hydroxyl 3 prime hydroxyl group, and again 3 prime hydroxyl group will be empty, and this chain will grow increasingly by means of DNA polymerase. And this formation of phosphodiester bond between 3 prime and 5 prime, 3 prime and 5 prime of the coming DNA nucleotide. This reaction needs energy, so energy is provided by these phosphate molecules which are present with every nucleotide. So gradually this chain will grow ahead. So up to this we are keeping our here, and from here we will explain one thing, and further we will study, we will continue in next class. So from up to this stage what we learned, that each bacterial DNA, each bacteria have DNA. When bacterial cell divides, DNA is going to be divided, and this division is uh, follows semi-conservative model. And what is semi-conservative model? In semi-conservative model, there is one daughter strain, one parental strain, which works as a template, and here daughter strain is going gradually. How daughter strain is going? How daughter strain is going? It is growing from 5 prime to 3 prime. It is 3 prime is empty, where 5 prime of the coming nucleotide will bind with antiparallel nucleotide, and chain will grow. So for this you can go through book, and you can discuss with colleagues, and you can ask from me to understand this thing in